Hello, welcome back to my channel. The topic of my project is identification of brain tumor initiating cells. Objective. Our objective has three goals. First, to target the root cause by eliminating brain tumor initiating cells, which drive recurrence. Second, to create better lab models of brain tumors for accurate drug testing. And third, to develop personalized treatments using BTIC data, ensuring more effective patient-focused care. Research flowchart. The flowchart outlines differentiating brain tumor from healthy brain images using pre-trained BGG16 and SVM. It involves data access, preparation, encoding, normalization, splitting, model training, evaluation, test data, predictions, and calculating performance matrices, confusion matrix accuracy to differentiate brain tumor images. Start access dataset, capturing trend data as well as label to their respective list, conversion of from list to arrays, perform encoding, perform split operation, performed normalization operation, demonstrated one encoding separation, consideration of pre-trained VGG16, extracted features to SVM classifier, provided training to model via training set, considered test data to evaluate performance of model, predicted responses from test data, Deloitte inverse Lie transform to retain original position of levels, import skill grid metric to calculate accuracy, calculation of confusion matrix for obtaining relevant performance matrices, differentiation of brain tumor images from healthy brain. Finally, end. Results and calculations. Finding the frequency of stem cells. Imagine we want to find out how many muscle cells are in a large group of cells. To do this, scientists use a special method called the extreme limiting dilution assay. They take a small sample and see how many stem cell communities grow when the cells are spread out at different densities. A computer program is then used to analyze the results and calculate the exact frequency of stem cells in the original sample. Determining statistical significance. When scientists test a new treatment or theory, they need to know if the results are a fluke or genuinely meaningful. That's why statistical tests like the t-test and ANOVA come in. For example, when comparing two groups like special ALDH plus cells versus regular ALDH cells, like tests show whether any observed differences are significant. If the p-value is less than 0.05, it means there is less than a 5% chance of the result happens by the random chance, making the finding statistically significant. Future scope. Developing an in vitro model. Determining what specific cellular or molecular mechanism we want to know about. Choose from 2D cell cultures, complex 3D models, or organ on a chip system, depending on the complexity needed to mimic the in vivo environment. Acquire relevant cells and establish a culture to grow them in a controlled lab environment. Expose the culture cell to the substance or condition being investigated, such as a drug. Measure the analyzed cellular responses to understanding the drug's impact at a cellular level. Developing an in vivo model. Select an animal like a mouse or a rat that shares significant biological similarities with human and is appropriate for the disease or process being studied. Administer the drug or the other invention to the animal. Observe how many drug affects the entire organism, including its metabolism, toxicity, and overall efficiency. Measure drug concentration in plasma, assess absorption, and evaluate psychological and physiological effects to understand how the drug performs in a complex biological system. Acknowledgement. I would like to sincerely acknowledge IM Hills, the Department of Basic Science and Humanities, and the IM group for giving me this golden opportunity. Their valuable guidance and continuous motivation throughout the course of this research work, their encouragement, resources, and collaborative environment have significantly contributed to successful completion of this study. We are deeply grateful for their role in enhancing the quality and impact of our research. Thank you.